Hello students, we are studying Chapter 5 Morphology of Flowering Plants from the Unit 2, that is Structural Organization in Organisms. In my last video, we are studying the parts of the flower. We have seen that we are having four main parts of the flower: the calyx, corolla, androecium and gynoecium. Calyx were going to be the sepals, green color part present below the petals. The corolla is going to be the brightly colored petals are known as the corolla. And the male reproductive structure is going to be the androecium, which consists of the anther and the filament. So we have studied this in the last video together with the important part, that is estivation, which is the arrangement of the petals and sepals on the floral bud. Today we are starting with another important part here, and that is the gynoecium. So the gynoecium is going to be the female reproductive part of the flower and is going to be made up of one or more carpels. So what is a carpel? The gynoecium, or also known as the pistil, is going to be consisting of three main parts: stigma, style, ovary. This top portion wherein the pollen grains will attack from the male or the attach from the male is known as stigma. This tube-like structure will be called as the style, and the below portion which consists of the eggs is going to be, or the ovules, is going to be called as the ovary. Within the ovary, the ovules or the mother cells or the megasporangia are present. So in this manner, one stigma, one style and one ovary together will make your carpel. And such carpels is going to be your gynoecium. Now, coming to the three parts of this gynoecium, or three parts of the carpel, you can say. First is the stigma. It is at the tip of the style and is the receptive surface for pollen grains. So pollen grains will come and attach to the stigma surface. Style, it is an elongated tube that connects to the ovary. So this is going to be the elongated tube which will connect the stigma to the ovary. It is known as the style. The third part is going to be the ovary. Ovary is the enlarged basal portion on which the style lies. So on this part is going to be called as your ovary. And this ovary is going to be having one or more ovules attached to a flat cushion-like structure known as the placenta. So this is going to be the placenta to which your ovules are going to be attached in the ovary. So this stigma, style and ovary are making your carpel. Now, the carpels can be of two types: apocarpus and syncarpus. So pistils with many carpels are going to be of two types, that is apocarpus and syncarpus. Apocarpus, when carpels are free, like that in the case of your lotus or rose, the carpels are free. They are known as the apocarpus. Syncarpus, when the carpels are fused, like you can see in the mustard and tomato, they are going to be called as the syncarpus. Now, coming to the important part over here, like estivation, again important part in this lesson is the placentation. One of these is going to be asked into your exam. It is a very frequently asked question, placentation. So placentation is nothing but it is the arrangement of the ovules on the placenta within the ovary. So here we are talking about the ovary. So in the ovary, how the ovules are attached, that is going to be called as placentation. So arrangement of the ovules on the ovary. So arrangement of the ovules on the placenta within the ovary is going to be called as the placentation. There are 5 main important types of placentation: marginal placentation, axile placentation, parietal placentation, and free central, and last one is the basal placentation. These are going to be the 5 main types of placentation. Let us study each one of them in detail. Here, the first is the marginal placentation. So, the placenta forms a ridge along the ventral structure of the ovary, and the ovules are born on this ridge forming 2 rows. So, you can observe in this diagram, the placenta is going to be present at the margin and the ovules are attached on this row forming two on both the sides. You can see that the ovules will be attached. So, this type of the marginal placentation you can commonly see in your pea plants. So, this is going to be the marginal placentation. Axile placentation. Here, the placenta is axile and the ovules are attached to it in a multilocular ovary. So, multilocular means here we are going to be having many chambers or locules in the ovary. You can see over here how the placenta has got divided in such a manner that the cavities will be formed in your fruit or in the ovary. So, here the placenta is axile and the ovules are attached to it in a multilocular ovary, many chambered ovary. You can observe this type in the plants like China rose or simple like tomato, lemon. You can find these type of the ovaries. This is known as the axile placentation, in which multilocular ovary is there. Coming to the third one, that is a parietal gynoecium or parietal placentation. Here the ovules develop on the inner wall of the ovary on the peripheral part. So, here the chambers are not present, but the placenta is going to be covering or surrounding the ovary and the ovules are going to be present hanging to this placenta. So, ovary is going to be one chambered, but it becomes two chambered due to the formation of the false septums. Septums is nothing but the partition. That partition is false because we don't have exactly the placenta, but the seeds are going to be forming somewhat distinctions. So, here the ovules are going to be developed on the inner wall of the ovary or on the peripheral region, as you can see over here. This type of placentation you can see in the case of your mustard, that is your mohri what we call, and ergemone. The next is going to be the free central type of placentation. So, what is free central type of placentation? The free central, here the ovules are born on the central axis and the septa are absent. So, there is no cavity or anything. The placenta is present in middle, long placenta which is going to which the ovules are attached. Simplest example of the free central is going to be your chili. You can observe in the chili plant or in the prime rows, you can see the free central type of placentation. The last one is going to be the basal placentation. Here the placenta develops at one side, that is at one base of the ovary, and only a single ovule is attached to it. You can see this type of the placentation in the flowers like sunflower and marigold. Marigold is your jhandu what we call. So, here there is going to be only one ovary attached at the base. So, hence it is going to be called as the basal type of placentation. So, here placenta develops at the base of the ovary and a single ovule is attached to it. So, that is basal placentation. So, these are going to be the 5 types of placentation. So, what is placentation? Arrangement of the ovules on the placenta within the ovary is going to be called as placentation. Which are the 5 types of placentation? They are marginal placentation, axial placentation, parietal placentation, free central and basal are going to be the 5 types of placentation. Very important from the exam point of view, that is aestivation and placentation. So here in a summary we can say that after fertilization, once these ovules are getting fertilized by the pollen grains, the male gamete is coming. We have seen the reproduction in angiosperms. So once all the reproduction events are over, the ovules that are there are developing into the seeds. So observe here, in this case we can see these are nothing but the ovules of the flower which are developing into the seed. And the ovary that is going to be there will mature into a fruit. And hence after fertilization, the ovules develop into seed and the ovary matures into a fruit. So this is going to be all about the flower, the types of the flower, the parts of the flower, aestivation and placentation. So this is what there into your cut-off syllabus. Many of you are asking me how much portion from this lesson is there. In this lesson, what is included for this year according to the PU board is the flower, the inflorescence and the flower. So whatever we have studied in my last 3 videos starting from the inflorescence until now is going to be there for your exam. We are not sure about how much will be retained or cut down, and so I have taken this complete lesson so that it will be also helpful for you into your competitive exam like the CET and the NEET. So because of that, I have not left any part from this lesson, as it is also forming a base for what you will be studying into your PUC second year first lesson itself. But from your exam point of view, this year you will be studying, or what is important is going to be the inflorescence and the flower. After this, there is a short part we will finish just in brief. It will not be there for your exam. The fruit and the seed. So what is the fruit? The fruit is nothing but the ripened ovary developed after fertilization is the fruit. So it is a characteristic feature only of the angiosperms, that is the flowering plants. Only the angiosperms are able to produce the fruit. A fruit that is going to be formed without fertilization. So in all the cases fertilization is not possible. So that fruit which is formed without fertilization is called as the parthenocarpic fruit, important in CET. Then in plants like mango and coconut, fruit is called a drupe fruit because they are going to be one-seeded and develop from a monocarpellary superior ovary. Then when you are talking about the part of the fruit, here we can see it is having a fruit wall which can be fleshy or dry, and it is going to be having the seed. So whatever this fleshy and dry thing is going to be there can be divided into 3 parts. That is, the pericarp can be divided into 3 parts: epicarp, mesocarp and endocarp. Epicarp is the outermost part, mesocarp is the middle part and endocarp is the inner part. Not necessarily the mesocarp is the edible part or you can eat it. In the mango you can see that the outer layer is the epicarp, the fruit that you are eating is the mesocarp, and the seed is going to be present covered by the endocarp. But the same thing in the coconut you can see that the outermost part that is going to be there, this is going to be the epicarp, this fiber is going to be the mesocarp, and the endocarp is going to be covering your coconut. So in this you are not able to eat the fruit, but instead you are eating the seed or the seed endosperm. So in different fruits the edible parts are different. So in this manner, this is going to be a short thing about the fruit Coming to the seed, it is the mature ovule developed after fertilization is going to be called the seed. A seed is made up of the seed coat and an embryo. So outer seed coat it is going to be having, and within that the embryo is present. Embryo is made up of the radicle, that embryonal axis, and one or two cotyledons. So what are cotyledons? We have seen these embryonic leaves are nothing but the cotyledons. In fact, based upon this only, there are two types of plants, that is monocotyledons and dicotyledons. Monocotyledons, example your meat and maize, here you can observe they are having only one single cotyledon. So they are going to be called as the monocotyledon. Here two embryonic leaves, so dicotyledon, example in the gram and the pea. So in this we can see that the outermost seed coat is going to be having two layers, that is going to be the testa and the tegma. Then the outermost is going to be the testa and the inner layer is the tegma. On the seed coat you can see that there is going to be a scar. Scar is nothing but a mark which is going to be called as the helium, through which the seed is going to be attached to the fruit, and above the helium there is a small pore known as the micropyle. Through this the water and the necessary nutrients are going to be coming inside and out of the fruit. From this seed only we can see it is going to be consisting of the radicle and the plumule. Radicle from which the root will develop, and plumule from which the shoot is going to be developing. So this is going to be all about the seed. So this much is going to be enough for you for this year. In detail I don't think you will have to study the structure of the monocot and the dicot seed, because in detail the structure of this is going to be there for you into your second year. So with this we finished your lesson, that is morphology of the flowering plants.